You know, when the group was up here and they said they're thinking about something fun, I mean, I was thinking about like the synthetic crop that would have the make the potato chips, spit out the beer, moonshine, whatever. So anyway, in moving towards that, uh, we figure the easiest way to get into synthetic plant genomes is through the chloroplast genome. The chloroplast genome, like mitochondrial genomes, are accessories to the nuclear genome. And so we've started doing this, and now we've realized that maybe we need to think smaller to go big. So the mini-Symplastomes. The whoops. The tobacco is the plant that's been genetically engineered the most, probably, especially for chloroplast engineering. So just popping in single genes, right? So we chose that as our first plant to go for, and not for smoking. It's just a model, right? But the plastome, the native plastome, tobacco genome is about 150 kb, so it's reasonable in size. It's got dozens of genes, and these genes have a very much prokaryotic type of structure. So why the Symplastome? Well, functionally speaking, it has a lot of benefits. One is that you can really express transgenes to a very high level. You can also get coordinated expression because you can use operons, just like a bacterial system. There's maternal inheritance, which means that the transgenes, the Symplastomes, anything that you put on the plastome, basically wouldn't be spread through pollen. And there's a lack of transgene position effects because you're just making the whole thing and sticking it in. So you only need one transgenic event. So what we're doing is we're shooting for fuels and bioproducts, and ARPA-E's funded that, started last year. We're shooting for improved photosynthesis, basically installing carboxysomes in an algal system. DARPA's funding that starting next month. And the one I'm really excited about is nitrogen fixation, because higher photosynthesis and endowing nitrogen fixation are probably two of the biggest sustainability traits. And I believe if you actually do something like that, people aren't going to care whether it's a GMO or not. If it has huge upsides like sustainability and can produce beer directly. So what we've done is we've designed and had synthesized various parts, and these parts are being and basically it's modeled after the tobacco plastome, but there's lots of changes in there. And so what we're hoping to do is to put it all together. In fact, we're assembling it now, and what we've noticed is that, of course, whenever you do something that hasn't been done before, there are problems and challenges. So some of the challenges are at the assembly level. There's some homology within the parts. Okay, so we're not sure that when they're going together at this point, they're going together exactly how we want them to go together. We're also worried about delivering the entire plastome, 150 kb thing, into chloroplast, and then replacing the native plastome. And then, of course, at the molecular level, it can be recombination between the Symplastome and the native plastome. So we started looking at some other systems, and so we've been inspired by algae, especially dinoflagellates, which are ancient eukaryotic algae that basically have mini circles instead of one giant plastome. So we've basically looked at that model of dinoflagellates and thought to build 10 mini-Symplastomes, all with a similar core, just like the dinoflagellate does it, and then blast all those in and basically to do co-transformation using the same selectable marker, various fluorescent protein markers, and then plug and play genes that come with the tobacco plant, but also things that we want to put in those mini Symplastome modules. So That's kind of the pitch. I appreciate your attention, and I believe there's alcohol over there. Thanks.